As antibody modalities grow more complex, traditional discovery workflows often fall short in predicting manufacturability. Transient expression systems can miss architecture-driven risks such as chain imbalance, mispairing, instability, and aggregation, allowing liabilities to emerge late in cell line development and CMC. In today's session, Raja Srinivas, co-founder of Asimov, will introduce RapidPools, a platform that bridges discovery and CMC by enabling high-throughput generation of stable CHO expression pools. By delivering early CLD-relevant expression and quality data, RapidPools allows protein engineers to assess manufacturability during lead optimization, improving lead selection, and reducing downstream risk. Raja will share real-world case studies illustrating how stable pool data revealed gaps between transient and stable performance and enabled targeted redesign to rescue challenging molecules, ultimately expanding the viable design space for next-generation antibody therapeutics. I'm your host, James Crosspayne with Bioprocess Online. I'd like to thank you, the audience, for attending today's event. This is live, so we do encourage your participation. Take a moment to get comfortable with the tools on your screen. I'd like to draw your attention to the question box in particular. Please share your thoughts with us during the presentation. Raja will answer as many questions as possible, and any we miss, I'll pass along for follow-up after the event. This question box can connect you with me if you run into any trouble, but please try refreshing your browser first, as it will fix most common issues. Finally, today's event will be available on demand. You'll receive an email with a shareable link shortly after we conclude. Now, without further ado, let's begin today's presentation. Welcome, Raja.
Thank you so much, and I'm really excited to talk to you guys today about some really new cool tech we're building to bridge discovery and CMC with an offering we call RapidPools. So as an overview of the presentation and the webinar, we'll go into 6 parts. I'll first give a high-level Asimov overview, then I'll set up the problem statement, talk about our solution in the form of RapidPools, go through 2 case studies, and then wrap up with a summary. So very quickly, the Asimov overview. We are a tools and services company building a tech stack to enable better bioproduction of therapeutics and the design of therapeutics. We are located in the Boston area, raised a little over $200 million of venture funding from the likes of Canada Pension Plan, Fidelity, etc., and are 100 people. The tech that we're building is an integration of biology and software. So on the right, I'm showing you the biology part of the tech stack in the form of cell lines that we engineer along with genetic parts and systems. That's then combined at a very deep level with an entire set of models and computational software to enable the design of genetic systems and bioprocesses to optimize bioproduction systems. So the tech that we're building is an integration of cells, genes, models, and software, all working in concert to optimize various attributes for bioproduction, titer, quality, timeline. So the way we work with companies is twofold. The first is we can license the platform out. That's typically done to big pharma. We call it the tech out licensing business model. And the second is where we provide services, cell line development, process development, and analytical development services at our Boston site. To date, we have over 50 biopharma partners spanning large pharma, mid-sized biotech, and virtual companies, along with CDMOs. And we've worked on over 70 drug programs to date, spanning monoclonal antibodies, bispecifics, gene therapies, etc. So today I'll focus on our biologic system, but we are currently also commercializing production systems for a few other application areas, including lentivirus and AAV. In those spaces, we're building stable systems for production of lenti and AAV in hepatocyte 3 cells, and we're going to be launching an RNA platform in the coming months. But today we'll focus on the CHO system. Before we do that, I wanted to just talk about CHO Edge. And the CHO Edge system is we're building a holistic solution across the entire therapeutic journey. Today we'll talk about RapidPools, but we are of course also having an offering in cell line development. we do offer a system for process development, and we're soon launching a offering for therapeutics development. The idea for the CHO-Edge platform is to allow developers the best-in-class tools across the entire therapeutics journey. And we'll touch much more on that in the coming slides. So now we're going to set up the problem statement and what we're trying to solve with RapidPools. So currently, therapeutic developers typically use transient production through the entire discovery journey. So what I'm showing you here on the left is the journey a developer goes through from antibody discovery through lead optimization and finally to cell line development where CMC begins. And typically what happens is a number of lead candidates, it goes down as you go from the discovery stage in the thousands all the way to cell line development where you might have one candidate to take forward for CMC and eventually clinic. The workflow to produce these therapeutics in the discovery usually leverages transient systems. Those transient systems are then used to make all the decisions to lock the lead candidate, and that final sequence is sent to cell line development where stable clones are made. And therein lies the problem we're trying to solve, is that there is a discovery and CMC disconnect. The systems that are used to make material in discovery is not the same as what's used in CMC and GMP production. At the worst, the challenge there is threefold in our opinion. Number one, and most importantly, is translation risk. And I'll come back to the idea of risk and de-risking throughout the presentation, which is poor correlation between titer and product quality in transient and stable introduces a lot of risk when you're going for a development of your product of your therapeutic. Second, it's slow. When you transition to CLD, you have to do a bunch of redesign, plasmid redesign, pool generation, etc. And finally, transient systems are notoriously high variance in terms of productivity. And so it can get very costly to make material for early stage and late stage animal trials. And so one thing we did at Asimov is trying to understand, are transient and stable systems have a high correlation? So we did a very simple experiment where we looked across 9 approved monoclonal antibodies, so a dozen monoclonal approved antibodies, and just looked at the transient day 3 titer on the x-axis and the stable day 7 titer on the y-axis. And what you can see is that there's almost no correlation between the stable titer and the transient titer. That can be visualized also by looking at the slope between the stable and the transient, and the slope is almost near 0. And this is just for monoclonal antibodies. This gets much more complex when you go to these more unique modalities like bispecifics, etc. And so this issue, what we're highlighting is that there's a lot of architectural driven liabilities, like the chain balance, the assembly within the cell, and the product quality that are not revealed in transient systems. And this can delay the development timeline by up to 1 year. And that's the problem we're trying to address with RapidPools. And so I wanted to just do a quick comparison of the way people produce with transient all the way to stable across a few dimensions. And the metrics we're looking at on the bottom here are the translatability to GMP product, the titers, the cost, reproducibility, the speed, and the throughput. It's a lot there, and I won't talk about each one, but you can see the chevrons trying to depict how we think about these systems in terms of those attributes. Transient systems typically are produced in hectare 3 cells, sometimes CHO-S cells, and they're typically done with a single gene vector, no selection, transiently. And if you have multiple chains in your molecule, you might use multiple plasmids with a dual transfection or a triple transfection. Now, the advantage of transient is that they're very fast and very high throughput. That's indeed why they've been used for so long to produce material. But the real limitation comes in the translatability, the titers, the cost, and reproducibility. On the other end altogether is the final stable clone. Typically in a CHO-K1 cell line or CHO-DG44 with metabolic selection, you have dual gene vectors. The integration can be via a transposase. And for those, of course, that is the most translatable to GMP product. It's what's used to make GMP product. But the big drawback there, of course, is speed and throughput. So the intermediary is stable pools, traditional stable pools, which if used with the right integration technology are fairly translatable to GMP product, can be high titer and low cost. But the challenge is that stable pools are very slow and not low throughput. And that's what we saw with RapidPools, where now we get all the benefits of traditional stable pools, but with the speed and throughput that you need to look at multiple candidates. And so now you've gotten a very nice bridge between the discovery stage and the CMC stage. So now I want to lay out the technology and what the RapidPools entails. And so going back to the diagram I showed a few slides ago, RapidPools sits in the lead optimization stage. So to be clear, we still envision developers using transient systems at the very early stage of antibody discovery. But once you have a set of candidates and are doing lead optimization, we think that's the right time to go into stable pools, in this case, RapidPools. And then that goes directly from discovery to CMC, and that bridge is now drawn. And so what that leads to, in our opinion, is three key things. It's de-risking lead selection, because now you know the CMC attributes coming out at the discovery stage before you nominate your final lead. It reduces time to market. I'll have a timeline slide in a few minutes. And then finally, it's improved economics. These stable pools are typically more performant than transient systems, which means you can unlock higher yields and more consistent scale up. So having set that up, what does a tech stack entail? And it's really 4 key pillars. And what comes in as the input are binder sequences, therapeutic architectures, and an optimization goal. I'll come back to optimization goal later, but what I mean by that is, do you care just about titer or other attributes as well? And so the platform is 4 key elements all coming together in concert to enable the RapidPools offering. The first is a host cell line. The second is transposase technology. The third are vector optimization tech. And then finally is the workflows. I'll talk first about the host cell line, then the transposase, then vector tech, and then finally the workflows. And the output then is stable pool RCBs for multiple lead candidates, along with purified material and analytical panels. And then the top clone or the top candidate can be cloned out for IND enabling studies. So first we'll talk about the host cell line. So the host cell line has a very nice provenance. So I'm showing you here from left to right the provenance, the performance, and our track record. So from a provenance perspective, we start with a adherent CHO-K1 bank from ECAC. We adapted the cell line to suspension, knocked out the GS, GMP bank to the Charles River, and now we have multiple FDA filings, IND filings across jurisdictions. The cell line grows really well, as I'm showing you on this graph over here. It's been scaled up to 1,000 plus liters and leverages off-the-shelf consumables. And we now have a regulatory track record in the form of INDs across jurisdictions. We also have a drug master file with the FDA for the cell line. So this cell line, which forms the basis of our platform for clone generation for GMP production, is now the exact same host that's being used for this RapidPools offering. The second part of the tech stack is a transposase. And the way a transposase works is it's a cut and paste mechanism to integrate vectors onto the genome. The transposase recognizes ITRs on the flanks of the vector and then integrates those on the genome in multiple copies independently, so you don't get concatemerization. And so what that means then is you get high expression, stable expression, and rapid recovery. At Asimov, we had a V1 transposase that was Sleeping Beauty class, and we've since now computationally mined a second transposase that's now productionalized and in operations for client campaigns. And I want to just compare the two transposases because we think that this new transposase is really critical as an unlock for this RapidPools. So over here is the data showing the legacy transposase and the current transposase. The dots are the legacy, the diamond is the Asimov transposase across 4 molecules. Critically, the new transposase is much faster recovery time compared to the legacy transposase. We're going in some cases from a 20-day recovery to a 10-day recovery, for example, for the fusion protein. That's a large unlock because when you're making stable pools for multiple candidates, you want to go as fast as possible. Secondly, you want to get higher titer, and in this case, on average, we're seeing about a 35% increase in titer. So the transposase is the second part of the tech stack. So the host cell line, the transposase. The third is model-guided expression optimization. And so we think a lot about the architectures that come out for various types of molecules, bispecifics, trispecifics, et cetera. And for every single molecule, the thesis at Asimov is that there's no one-size-fits-all vector. You want to optimize vector expression on a per molecule basis. And so we built a set of models and tools to enable that across the plasmid design. So what are the expression elements that go around the plasmid to express the GOI, signal peptide design, and then finally codon optimization. So let me just take you through those 3 things really quickly. Vector designer, signal peptide selector, and then the codon optimization. So on the signal peptide, on the vector designer, what we've developed at Asimov is a set of genetic parts, thousands of genetic parts that have been experimentally characterized in the cell line of interest. So for example, in blue here, I'm showing you a library of promoters, some being very strong to some being much weaker. And you might ask the question, why do you want a weak promoter? And the answer could be to attenuate the GS, the selection stringency. So across every single part of the biologic expression plasmid, we have a set of parts that can then be mixed and matched to build bespoke vectors. Now with that parts library, the combinatorics explodes. It literally is trillions, if not more, combinations that you can use to build these expression vectors. So you need a way to rationally search that space. And we do that in kind of a couple of ways. Number one is via architecture-based vector templates, and the second is via simulation. On simulation, real quick, because we have quantitative data on every single one of these parts, we can understand how these various combinations might work in silico before we build them in the lab. And so we're now able to go from a very, very large design space to a much more manageable design space via these templates and these simulations. So now what we have is we have all the elements that go around the plasmid. The next step is a signal peptide. And so at Asimov, what we've done is we mined over 3,000 signal peptides. That's been across 200 biologic data sets and experimentally validated a handful of them. So the question, of course, is does signal peptides matter in terms of per molecule basis? And here's some data to kind of try to convince you that it does. On the x-axis is 5 different molecule types, different architectures from different campaigns. On the y-axis is the relative titer compared to a baseline signal peptide. That signal peptide is actually one that is more performant than ones that are used in the literature. And what you can see is that every single color is a different signal peptide. And each bar is the optimal signal peptide that we found for every single molecule. And what you can see is that there's signal peptides can make a very big difference in terms of titer. So for example, in one case, we're over 6-fold improvement in titer compared to the baseline signal peptide. And that number does bounce around, but it does go to show the importance of signal peptides. So we then use our computationally mined signal peptides to pick one that is fit for the molecule. So now we have the expression vector and the signal peptide. The final step is turning the amino acid sequence into the codons, the CDS. And so to do that, we've built our own algorithm, and our algorithm is a kind of a model that's trained on the CHO genome statistics, which we then further refine with various biophysical attributes. And what you can see here is we compared this to what we think is the best third-party codon optimizer. So the data is not shown here, but at Asimov a few years ago, we did a thorough benchmark of all third-party optimizers. And what we found was a very large distribution in terms of the titers and the expression from the different optimizers. And so we built our own, and we benchmark against the best third party. And so in some cases, we are, quite performant compared to the best third party, seeing, up to threefold gains in titer. In some cases, it's much more modest. It's more like a 10% gain. In some cases, in the middle, 30%, a doubling, etc. So what we believe we have here is a really strong codon optimizer that pairs nicely with the signal peptide predictor and the vector designer. So what comes out of this entire data-driven vector design is a very thorough exploration of vector design space, and we use computational models to guide every step of that journey. And then finally, we've now developed an entire set of processes to enable high throughput stable pool generation, to allow hundreds of stable pools using lab automation and miniaturization at COD grade. And I'll come back to that in 1 minute. But our initial workflows were at lab scale, and now we've miniaturized that, which enables this RapidPools offering for us to be able to generate pools for many candidates at the same time. So that's the tech stack. It's the host cell line, the transposase, the vector design tools, and then the processes for high throughput pool generation. Before I go through the two case studies, I want to talk about timelines. And the timeline I'm going to talk about is not transfection to RCB like we talk about in cell line development, but lead optimization to RCB. The typical timeline, even with a very strong cell line development system, is about 29 weeks. So to walk through that timeline really quickly, initially on the candidate selection side, there's a DNA design stage. And then typically there's transient material generation. It might be 2 rounds of material generation to get enough material across 2 screens. The candidate gets locked in. That amino acid sequence gets shipped over to CLD, where another DNA design workflow is kicked off to make vectors for that CLD campaign. Then you make the stable pools, the clones, and then you get the RCB. So all in, from week 1 of the candidate selection to the end is about 29 weeks. Now, when we look at the RapidPools, we're able to save 7 weeks off of that journey. And how does that work? It works in 2 key ways. The first and foremost is that the pools that are being generated during the selection phase, the candidate selection phase, can be directly cloned out to cell line development. And that's shown here in green, where the candidate selection happens and the clone out starts right away. So what that means is that there's no rederiving of the DNA or making the pools again. You go straight to clone out. That's a very large savings. And the second is that because stable pools typically are more performant than transient, you can get the same amount of material with a 1-week production run compared to a 2-week production run. So that's another area for time savings. And so put together, what you see is a 22-week timeline from, again, lead optimization to RCB, to be very clear on what those endpoints are. And so the benefits of the RapidPools approach is reduce attrition rate. So again, reducing risk with rapid timelines, and then also with a lower cost. And so in our opinion, that's a real unlock for developers where time to market is critical for any given therapeutic campaign. And then finally, all of this is only relevant if you get high titers at the clone out stage. And here's just a few sample case studies from our cell line development suite across monoclonal antibodies and bispecific antibodies. We're routinely getting 8 to 12 grams per liter, and we see this number climbing year over year. And so with that, that's the technology overview that enables a RapidPools. the 4 pillars leading to very high clones at the very end of cell line development. And so now I want to go into the case study of how we de-risk candidate selection for a client. And so the context here is that this client came to us with multiple candidates. They had binders for 3 antigens they discovered during an antibody development campaign. And they took those 9 paratopes and made a set of 12 multi-specific candidates. And these candidates range from 2-chain bispecifics to 3-chain bispecific, to 3-chain trispecifics. And what they wanted to understand was which molecule to move forward for cell line development and ultimately to the clinic. And so what we did for them was use a RapidPool platform to help them make that decision versus just using transient material. So what we did was the input was these 12 amino acid sequences. We did an entire set of vector design using the tools I talked about in the previous section. And the output was over 70 genetic variants. We did 4 replicates to make a total of 288 stable pools. And what we did was we generated the pools, did a 7-day run, did Pro-A purification, and then did an analytical panel. And the metric we cared the most about was what was called effective yield, which was the titer multiplied by the heterodimer percentage. And here's the data for that. What you're seeing on the x-axis is the transient yield, and the y-axis is the effective yield from the stable platform. And what's striking is that there's almost no correlation between the transient titers and the effective yield that we measure. Visualize another way. The top pool that we rank, here we call T3, was only 3rd in transient. And there's really, this plot is in some sense all over the place. So what that means is that this particular client would have nominated a different molecule to take forward, one with much lower actual developability attributes compared to the one that they did take forward. And so here's that, where they then looked at the developability ranking that we provided them, along with some efficacy and safety data. And here that's visualized, by the various rankings that they did with their in vivo assays, low, medium, high, and high, in terms of how effective the molecule is. And then on the y-axis is the effective yield rank. And now they're pursuing a couple of molecules going to cell line development, and will choose one final one as they get the final animal data to choose their lead candidate. So what this case study shows is that the transient data is not faithful for what stable pools produce. And if they'd chosen just on transient, it'd have been a very different set of molecules, and those molecules would have had much lower effective yield metrics, and that's where they were taking much more risk going to the clinic. So this approach de-risked them from going from their final 12 molecules down to the final candidates for the clinic. So now I want to talk about a second case study around molecule rescue. So I teased this a bit at the beginning around how we're getting the therapeutic design, but we have done some work in that space. So to set the context up there, we worked with Amgen on a bispecific antibody rescue campaign. And so here's the context to set that up, which is they had 3 IgG scFv bispecifics. So these are 2-chain molecules. The challenge is very easily notable in the graph. Bispecific C needed rescuing. It was much lower titer compared to bispecific A and B. And they tried a bunch of different things to try to get the titers up, but it had not been successful. And so they came to Asimov to work with us on this. And now this case study is now published on bioRxiv, and that publication is now available. So what we did was we used a suite of computational models to come up with a set of hypotheses. And these hypotheses span the entire stack of the central dogma of biology, from the DNA level to the RNA level to the protein level, looking at various types of areas where bispecific C might have been malfunctioning compared to bispecific A and B. And what we found were 3 plausible failure modes. The first was splicing abnormalities. The second was this very interesting thing where there was a ribosome collision motif in the amino acid sequence. And the third was local codon liabilities. And one thing to note is that these molecules were fairly homologous, and there's just a few key differences in the amino acid sequence. So we did an initial screen for each of these hypotheses, and what we found was the splicing abnormalities could not explain the difference. But the RDK motif, the ribosome collision, and the local codon liabilities could. So we then put the two together and tried to understand what was contributing to the low expression from the ribosome collision versus the local codons. And what we found was it was additive. The ribosome collision was contributing about a twofold, and the codons were another fourfold. So we put the two together, we're seeing an 11-fold increase between the original sequence and the final sequence. And to be clear, the ribosome collision fix was an amino acid tweak. We changed the motif from an RDK, which houses the ribosome collision motif, to an RDN, which gets around that. So that amino acid shift allows us then to get the twofold gain in titer, and then fourfold more from the codons that we chose. So it's a very nice case study of how we can use computational and synthetic biology tools to rescue molecules. So in summary, what we're trying to build is the bridge between discovery and CMC. We see that candidates, especially as we get more complex molecules, there's more risk being underwritten from a manufacturing perspective, the titers, the product quality, et cetera, and that can have deleterious impacts on programs. We're trying to build that bridge in the form of RapidPools, where at the lead optimization stage, developers go to RapidPools compared to Transient, and then that pool can be advanced straight to CMC via cell line development and GMP production. And again, what that leads to is de-risk candidate selection, reducing time to market, and then of course, improved economics. And so with that, thank you so much for the opportunity to present this webinar. Happy to take questions.
Fantastic. Thank you so much, Raja, for the excellent presentation. Before the Q&A begins, I'd like to remind our audience you can connect directly with Raja. His information is located on the right side of your screen. We see a lot of great questions coming in. Thank you so much for that. Please keep submitting those via the Q&A box below me. We're going to address as many as we can, and the rest I'll pass to Raja and the Asimov team to follow up offline. Our first question, how many sequence variants can Asimov support with RapidPools?
Yeah, that's a great question. And in the case study I showed, that was 12 unique amino acid variants. We are now talking to various companies around 50, 60, 70, even up to 100 variants, amino acid variants. And then we also can explore vector design space for every single amino acid variant.
Fantastic. Our next question, can you produce material for research and development work?
Absolutely. And so that's the one thing that, again, in this case study we did was we produced material, using a 7-day batch. That was just for analytical, but we have the capability to produce material for both analytical development, but then also for in vitro and in vivo work to de-risk candidate selection.
Excellent. Our next question, can these pools go straight into clone out, or do they have to be rederived?
Yeah, that's a great question. And the offering that we're putting forth right now is the ability to go straight from the rapid pool that we generate in the miniaturized format straight to clone out. And that is the big unlock in terms of timeline savings, because it's no longer a vector redesign or stable pool, making a new stable pool.
Great. Our next question, do you measure just titer at the RapidPools stage?
No, we did not. Actually, that's a critical part of this, which is that with these more complex molecules, it's not just titer, but also the product quality attributes. So for example, in the case study I showed, we had a metric for effective yield, which was the titer multiplied by the heterodimer percentage. And that was what we care about at the end, because that's the final product that's going to be have the efficacy in the clinic. And so we will work to define that metric, which is usually some combination of titer along with some product quality attribute. And we have the analytical instrumentation at Asimov to measure those.
Excellent. All right, everyone, that's going to be all the time that we do have left for today. Don't worry if we didn't get to your question. I'm going to pass them along to our presenters to follow up offline. I'd like to thank Raja and Asimov for providing their expertise today, and you, the audience. We appreciate you being with us and hope to see you again soon. Have a wonderful rest of your day.